Hi students, this is A. Mandari Kumar Rajesh. From this video onwards, we are going to discuss examples of intergenic gene interaction. So today's video is about epistasis. Before we talk about epistasis, I want to share some important information regarding intergenic gene interaction. So the first point is, in each case, produced phenotype is distinct. In each case, produced phenotype is distinct. For example, capital W is a gene which is responsible for the character white color. Then capital Y is a gene which is responsible for the character yellow color. So the phenotype produced by the gene is very distinct. The phenotype produced by the gene is very distinct, very noticeable. Then the second point is, genes involving in this interaction shows normal dominance and assert independently. So genes involving in this interaction shows normal dominance and assert independently. That means genes involving in this interaction do not shows incomplete dominance or codominance. Genes involving in this interaction do not shows incomplete dominance or codominance. Then here the genes are able to assert independently. That means here there is no linkage between two genes. Here there is no linkage between two genes. That is why the genes can assert independently. So that is why the genes can assert independently. Then I am going to explain the third point. The third point is, on phenotypic level, homozygotes and heterozygotes will show their respective phenotype governed by the dominant allele. That means whether it is homozygote or heterozygous will show their respective phenotype based on their dominant allele. Then the fourth point is, F1 generation must be heterozygous, heterozygous for two gene pairs. So F1 generation must be heterozygous for two gene pair, that is capital A, capital B, small b. So these are the 4 points you have to consider in your mind while learning intergenic gene interaction. Now I am going to explain the phenomenon epistasis. Epistasis is a phenomenon in which a gene suppresses the phenotypic expression of a non-allelic gene is called epistasis. So a gene which suppresses the phenotypic expression of a non-allelic gene is called epistasis. Epistasis. The gene which suppresses the phenotypic expression of a non-allelic gene is called epistatic gene. Epistatic gene. While the gene whose effect is prevented or suppressed is called hypostatic gene. Hypostatic gene. Hypostatic gene means the genes whose effect is prevented or suppressed, that is called hypostatic gene. When these two genes are present together, the phenotypic character is determined by the epistatic gene only. The phenotypic character is determined by the epistatic gene only. Then epistasis reduces the number of phenotypes appeared in F2 generation of dihybrid cross. Epistasis reduce the number of phenotypes appear in F2 generation of dihybrid cross. That means in dihybrid cross in F2 generation we could observe 4 types of phenotypes. They are round yellow, round green, wrinkled yellow, wrinkled green. While in the case of epistasis, the number of phenotypes would be reduced. It may reduce into 3 or 2 or 1. So the epistasis reduces the number of phenotypes appear in F2 generation of dihybrid cross. Then epistasis can be divided into 3 types. They are dominant epistasis, recessive epistasis. The third one is dominant recessive epistasis. In this video, we are going to discuss the dominant epistasis. The dominant epistasis can be defined as the dominant gene at one locus suppresses the expression of another gene at different locus is called dominant epistasis. The dominant gene at one locus suppresses the expression of another gene at different locus. So I explain with the diagram. So this is two pairs of homologous chromosome. Here the alleles are fixed in this position. They are capital W, that is the gene which is present in this locus called capital W, and in this pair of homologous chromosome the genes are present in this locus, they are capital G, capital G. So here the capital W is the dominant one, is the epistatic gene which suppresses the expression of capital G, which they are present at different locus. So the capital W is present at one locus, it is able to suppresses the expression of the capital G which are present at different locus, that is called dominant epistasis. Then let me study the dominant epistasis in a plant called cucurbita pepo. Cucurbita pepo. Cucurbita pepo or summer squash or summer squash. Cucurbita pepo exhibit three colors. They are white, yellow, then green. The white color is the dominant one, that is the epistatic one. So white color is epistatic over the two other colors. So the capital W is the epistatic gene. Then yellow is dominant over the green color. Yellow is dominant over the green color. Then the yellow color is formed when only the epistatic gene is present in homozygous recessive condition. The yellow color is formed only when the epistatic gene is present in homozygous recessive condition. That means the hypostatic gene yellow, that is Y, is represented by the dominant allele. Now I write the genotype of that color yellow. So small w, here capital Y or small y. In this condition only the yellow color can expressed because of the absence of the dominant epistatic gene. Then the green color is formed when only the both epistatic gene and the hypostatic genes are in homozygous recessive condition. Then only the green color will be expressed. I repeat, the green color is expressed when both epistatic gene and hypostatic gene in homozygous recessive condition. Then only the green color will be expressed. Then I will write the genotype. So the genotype would be small w, small y. In this genotype only the green color will be expressed. While in the white color a single gene or allele capital W is enough to produce the white color. The single capital W allele is enough to produce the white color. Now I am going to figure out the dominant epistasis cross in cucurbito pepo. Now I am going to make a cross between pure breeding white summer squash with pure breeding green summer squash. So the genotype is capital W, capital W, capital G, capital G. So the phenotype is white. Here the genotype for the green summer squash is small w, small w, small g, small g. Then the phenotype of this individual is green. The produced gametes would be capital W, capital G. Here small w, small g. Then cross pollination takes place, results in the production of F1 generation. Then the F1 generation should be heterozygous in nature. So capital W, small w, capital G, small g. So it is a hybrid, F1 hybrid. It shows the phenotype white. It shows the phenotype white. So in cucurbito pepo, the fruit colors are three. They are white, green and yellow. Here the epistatic gene is present, so the fruit color is white. Then F1 generation is allowed to self-pollinate. So F1 generation cross with F1 generation, that means capital W, small w, capital G, small g cross with capital W, small w, capital G, small g. The cross is made between these two individuals to develop F2 generation. So the gametes produced would be capital W, capital G, capital W, small g, small w, capital G, small w, small g. The same condition will occur here. Small w, capital G, small w, small g. Then these gametes are plotted in the Punnett square. So capital G, capital W, capital G, capital W, small g, small w, capital G, small w, small g. Here capital W, capital G, capital W, small g, small w, capital G, small w, small g. Then the fusion of gametes takes place. Capital W, capital W, capital G results in white color because the epistatic gene is present. Then capital W, capital W, capital G, then the fruit color is white because of the presence of the epistatic gene W. Capital W, small w, capital G, fruit color is white. Capital W, small w, capital G, small g, fruit color is white. Capital W, capital G, small g, fruit color is white. Capital W, small g, fruit color is white. Capital W, small w, capital G, small g, fruit color is white. Capital W, small g, fruit color white. Capital W, small w, capital G, so white in color. Capital W, capital G, small g. So the fruit color will be white. Here small w, capital G, capital G. So here the epistatic gene is homozygous recessive condition. Here the epistatic gene is present in homozygous recessive condition. Then the hypostatic gene, that is the yellow color, the hypostatic gene is contains the dominant allele. That is why it exhibit yellow color. So the fruit color will be yellow. Then small w, capital G, small g. So here also the hypostatic dominant allele is present, so the fruit color will be yellow. Then capital W, capital G, small g, white in color. Capital W, small g, white in color. Here small w, capital G, small g. So the fruit color will be yellow in color. Then small w, small g, result in green color, because the epistatic gene and the hypostatic genes are in homozygous recessive condition. So the fruit color will be green in color. Then the phenotypic ratio would be 1, 2, 3, 4, 5, 6, 8, 10, 12. 12 is to 3 is to 1. 12 white color fruit, 3 yellow color fruit, then 1 green color fruit. So that's all about the dominant epistasis. I hope that information will helpful for you, and I hope you all enjoy this video. Please stay home and stay safe. Then if you have any doubt, please clarify with me. Then thank you for watching this video. Thank you.